What is the molecular basis of phenylketonuria? A presentation by Team 23.
The structure on the left is the wild-type phenylalanine hydroxylase structure. It can exist in the cell in either dimers or tetramers. Here we show a tetramer. Shown in the structure is 4 ligands, phenylalanines. 1 ligand is bound in each of the chains. On the right shown is a dimer of the PAH enzyme. Shen et al. Conducted a study of the different PKU mutations. Different PKU alleles were co-expressed, and the PAH enzyme activity levels were assayed. The 2 yellow arrows point to the sites of the 2 mutations we're focusing on, I65T and R408W. The genotypes constructed were compared to the predicted PAH levels on the PAH locus-specific database.
While there are multiple combinations of mutations outlined in the Shen study that result in various degrees of disease severity, we chose a combination that results in classic PKU, or the most severe classification. The R408W I65T genotype results in 15% residual PAH enzyme activity and 83.8% in individuals classified as classic PKU. The R408W is a mutation to a tryptophan in the catalytic domain, causing the C-alpha-13 C-beta-7 loop to be mostly buried. The I65T is a transition mutation to the regulatory domain, causing a disruption in the hydrophobic packing of the regulatory core. These mutations affect the PAH enzyme's ability to perform its metabolic function.
Moving on to changes in phenylalanine metabolization. Basically, PKU patients have a metabolic pathway failure. As we can see in the left figure, phenylalanine hydroxylase, PAH, and its cofactors BH4 and O2 convert phenylalanine into tyrosine in a healthy human liver. And on the right figure, we can see that amino acids are essential to create proteins for almost all physiological processes. Specifically, tyrosine is important for the production of many neurotransmitters in the brain, such as dopamine, epinephrine, and norepinephrine. And so PKU patients can't get the tyrosine precursor, and this can lead to many neurological problems. Also, phenylalanine buildup in the blood is toxic and can be fatal. And because of this, all babies are tested within 48 hours of birth for PKU with a heel prick. And lastly, some future research. The Williams group is attempting to insert a functional PAH into patients with PKU in attempts to stop phenylalanine buildup.
Patients with PKU tend to show high concentrations of phenylalanine in tissues and blood. Low concentrations of tyrosine can also be observed. The current hypothesis is that high levels of phenylalanine and compounds containing it act as neurotoxins in the brain, leading to severe neurological damage that can result in intellectual deficits and neurodegeneration. They can induce oxidative stress, which might cause an imbalance between reactive oxygen and nitrogen species and antioxidants, leading to damage to proteins, lipids, and DNA. As mentioned earlier, they may also impair neurotransmitter metabolism. A number of studies have shown that high levels of phenylalanine in the brain tend to also cause low neurotransmitter levels. A few other mechanisms have also been proposed, such as errors in bioenergetics and calcium homeostasis, but those have at least a little less evidence than the ones mentioned above. It could potentially be a mixture of multiple methods, though.